Hello, today I'm going to be talking about the basic membrane and neurophysiology, and this should go into everything you've already learned about the cell. So, as we get started, we can actually go into a quick review of the cell. What we're talking about is talking about the nucleus, where we have the genetic material. We have the cytoplasm, which is a cell fluid, and the cell membrane around it, which is going to be really important for this. Within the cytoplasm, we do have ribosomes, which are proteins that make other proteins. They are technically an enzyme. We have the cytoskeleton, which gives the cell its shape. And you'll notice I'm actually using one color, mainly because should all be review. We have the endoplasmic reticulum, which is a phospholipid bilayer inside. We have the rough portion, which has ribosomes attached to it. And we have the smooth. We also have the Golgi apparatus, which helps sort where the vacuoles that we make are going to go. We have vacuoles, and we also do have the mitochondria. Now, we've already talked about how you're making energy and how your cells use ATP for its fuel. It needs to make the ATP by breaking sugar, and we talked about all the metabolism associated with it. So, again, just to recap, we have glycolysis, which you actually take glucose, C6H12O6. We'll actually just put a little ring structure here to represent glucose. So you're going to take that and break it, and you're going to end up getting two pyruvates. I'll put two P's for two pyruvates. That's glycolysis, and if you notice, you're not putting any oxygen in there, so it's anaerobic. What you are doing is you're putting 2 ATP in and getting 4 ATP out for a net of 2 ATP. Now the two pyruvates will go to the mitochondria, where they will be further broken up, but before they go there, they are converted into acetyl-CoA, acetyl-coenzyme A. And in the citric acid cycle, you're going to go through pretty much the cycle, get NADH and FADH2, and the FADH and NADHs that you make are going to go to the electron transport chain and eventually take the hydrogens and give you water. As you fully break everything down, you get carbon dioxide and water as a byproduct. And so when you get to the electron transport chain is where you really use your oxygen. Prior to that, you're not taking in oxygen to break anything. So all of it is anaerobic except for the electron transport chain. Now, uh, how can things pass in and out of the cell? And there's two things that are important. One is the osmosis, and osmosis is based on the difference in osmolarity. You'll notice you see osmolarity here, osmosis. Osmolarity is technically how many grams per concentration of moles in a liter. So technically you can actually take the atomic number of something, convert that to grams, and if you put that in liters of water, that's how many particles would be in it. Some particles break apart, some particles do not, which if they break apart, they actually have a higher amount of osmoles because they will break apart into two. Basic diffusion is the other thing that allows things to pass through in and out. Now, as a cell membrane is lipophilic, what can cross through just the cell membrane has to be lipophilic, unless it can go through certain transmembrane pores, which you have a lot of. Now, in cells, you can use proteins to move things in and out. I mentioned this probably in class as facilitated diffusion, which pretty much uses transmembrane proteins to allow things through. And there's two types. There's the voltage gated and the ligand gated. The biggest difference between voltage and ligand gated is voltage, as the name implies, a voltage has to reach a certain level for that protein to open. A ligand gated needs to have some sort of messenger molecule or some sort of molecule to bind to it to cause it to open. And it can be very specific. So you may have a sodium channel that only allows sodium, but it will not necessarily allow sodium chloride to move together. You also have another thing that is cotransport, which what you do in cotransport, you have a high concentration of something well, we'll go with high concentration on one side of the membrane and something on low concentration on the same side. And because of the high concentration of one molecule, the other one can go through. Usually the co-transport I like to use is glucose, which I'll talk about a little bit later. Now you also have something called active transport. Active transport, as the name suggests, is it's active. So you need ATP for energy to bring things in or out of the cell. And so you can actually have different types of active transports. You can't have just active transport where you pump one thing out against or in against a concentration gradient. You can also do something called anti-transport. And in anti-transport, things move in different directions. So you use energy to move two things against a concentration gradient. This is actually really important because we have a protein called sodium potassium ATPase that pumps sodium against the concentration, potassium against it, and so it actually gets even higher concentrations on one side than the other. A active transport co-transport will break ATP and get two things to move at the same time. The difference is this time it can use ATP. Now I always tell you that there are certain things that are important to look at. One of them is to constantly look at the fluid, the composition of fluid. If you look here, you can see sodium extracellular, so outside the cell, is a lot higher than inside the cell, about 15 roughly times higher. And so based on diffusion, if a channel were to open, it would get sodium to move in. If you want to move sodium out, you have to actively pump it out. If you look at potassium, you'll notice 140, 140, and these numbers will change, by the way. Clinics usually have a range. And so if you open a potassium channel, passive, potassium will move to a lower concentration, so it'll go from inside to outside. Calcium, you see 2, and you'll see the 0.001. So you open that, and you can see how it's going to move in. Magnesium is going to be a little different. Chloride, you can see that if you open a chloride channel, obviously you're going to move here. Now, if you look at the charge, you'll notice that if you were to open a chloride channel, you also are going to make it go farther, pretty much become more negative. Bicarbonate, you can see how you have more bicarbonate on the outside. So if you open that, it's going to come in. That also tells you that the pH will be different inside the cell, outside the cell than inside. You're going to have a higher acid. This is a hydroxide, phosphate hydroxide. And so you can see how you have more phosphate hydroxide in here. And this does help act as a buffer. You'll notice you have two, and so hydrogens can bounce back and forth. And you have sulfate here, which, again, you have higher concentration outside than in, and so you can see how it's going to run. Now, to know what direction things are actually going to move, you really want to use something called the Nernst equation. Now, the Nernst equation just tells you what the diffusion potential is on something, and it's based on the chemical electric equilibrium of something. So if you have more chemical electric aspect on the inside, then out it's going to be moving in. And this is actually the Nernst equation here. It's the E, it is an energy. You have a constant here, you have a gas constant, you have the normal temperature, you have the ion charge, Faraday's constant, and then the concentration inside and outside, and it's on a log-based scale. And so what happens is you can actually tell how much charge this is going to cause. So if we actually use this equation and we take the average human body temperature being 37 degrees Celsius, and I'm actually always going to use Celsius in here, even though in physics and chemistry a lot of times you'll use Kelvin. In biology we tend to use Celsius, which the conversion is relatively easy. And so we can actually do that. In this case, we are going to convert the temperature to Kelvin. And we know that sodium has a extracellular concentration of 140, an inner concentration of 10. And so when we plug it into the equation, what we can see is that sodium has a positive 70 millivolts, and so you're going to be trying to get that to move into the cell. I actually gave you the equation here and why we changed different constants. Now, as we are dealing with human body, you can use the T, the temperature, in kelvins here. So you can actually, even if you want, you can bring this down. You can look at the concentrations and bring that away. And what it will do is it will actually end up giving you an even easier equation to deal with. Now, everything we're dealing with is due to ion differences in a cell and the membrane. And as you have that, you do end up getting a charge. We call that charge the resting membrane potential. This is mainly due to a special protein that I mentioned before called the sodium Na, potassium K ATPase. This is going to break one ATP, and when it breaks an ATP, you're going to pump 3 sodium out and 2 potassium are going to be pumped in. This is actually going to be the main cause of the cell membrane, but it's also due to the other ions that are there and the pH. Acid levels, the pH levels on both sides of the cell membrane can actually cause a charge to form. It actually acts like a cell battery. Now, we also have something called an action potential. An action potential is how a cell will lose a charge and regain it. Now, one thing to remember that at rest, your cell, it doesn't mean your cell is not doing anything. It's actually preparing to get everything done. At rest, you have the resting membrane potential. What it means is you have a charge on the membrane because of the differences in ions. Again, remember, it's mainly due to sodium and potassium concentrations. And so if you've been pumping 3 potassium out, 2 potassium in, you have a charge because sodium, pretty much you've been pumping 3 positives out, 2 positives in, so you have a negative charge actually developed. Now this is a potential, which means you don't necessarily see this if you're touching it. It would be actually if you could look at the inside versus the outside. Once you actually open a channel, you do get the electrical charge move and you lose the positive charge. So if a sodium channel opens, we'll talk about in particular, a ligand-gated sodium channel opens, it would trigger a sodium channel to open, and the sodium would rush in, the sodium would rush in, and that would cause you to lose your charge. So we call that a depolarization, meaning the membrane no longer has a polar aspect to it. It no longer has a charge. Once this starts reaching 0, the sodium channels begin to close and the voltage-gated potassium channels open. Now, in reality, you do also have voltage-gated sodium channels, which I'll talk about later, which allow this to occur. And so what you have is you have the potassium leaves the cell and the cell begins to repolarize. And in some cases, it can hyperpolarize, meaning it actually becomes more negative. And then when you're at rest again, the sodium potassium ATPase returns the resting membrane charge. So one thing to remember is this, and I took this picture, you have the citation here from the article. You can see the minus 70 millivolts here. This is actually done because of the sodium potassium ATPase that opens. You have something called threshold in neurons in particular. And what happens is you can start raising the cell membrane potential, make it a little more and more positive by ligand-gated channels. Once you get to threshold, the voltage-gated channels open. If the voltage-gated channels open for sodium, sodium floods out and you get this depolarization. You notice that you have a 0 here. And so sodium channels close, but it continues to depolarize. And then potassium channels open and sodium channels close, and you end up getting the repolarization. So over here you have potassium, and you'll notice that I will use the letter Na. This would not be N, that's just nitrogen. It'd be Na for sodium and K for potassium. Once you get down to 0, potassium channels begin closing. It still leads to hyperpolarization. And sodium potassium ATPase starts working here, giving you a return to the resting membrane potential. The entire thing here, the depolarization and repolarization, is your action potential. And this action potential is specific for a neuron. A neuron is pretty much the functional unit of the nervous system. Now, I actually want you to know a little bit about some medications. And you have something like lidocaine, which blocks sodium channels. Now, if you think about it, if you block sodium channels, that means the voltage-gated ones. That means that even though you may actually end up getting to threshold, the voltage-gated sodium channels will not open. And if the voltage-gated channels don't open, you don't get the complete depolarization. Remember, it's variations in sodium concentrations will affect the depolarization. So if you have a hypernatremia, hyper means above, Na is for the sodium, and emia is blood, so you have high. Nature is the sodium in Latin, emia for blood, so you have high amounts of sodium in your body. You actually start getting certain symptoms. You actually can get excessive thirst. This is due to osmosis. If you have too much sodium, it will pull it out of the cells. They'll pull fluid from the cells out, and it will start getting you to feel thirsty. It can cause lethargy, and confusion and muscle twitching, and that has to do more with the neurons and actually even muscle tissues not being able to depolarize properly. So it changes how you depolarize. You drop this 140 too low, and the force and the speed that the sodium will come in will decrease, causing your nerves, your muscles, and any cells that have to depolarize to function less likely. Now, usually you don't see these last three unless it's severe. Main effects of hypernatremia tend to be more related to thirst. You also have hyponatremia. We talked about what nature means as sodium. Emia, we talked about it in blood, and hypo means below. So it means low sodium in blood, and this will actually end up causing nausea, vomiting, confusion, loss of energy, seizures, and coma. Again, it does have to do with, if this is too low, it will decrease the speed here. So over here, it increases the speed that it can go in, so it will end up causing twitching over here. In the hyponatremia, it will decrease the speed of the change. Now, we also can look at hyperkalemia. Now, hyperkalemia will actually show you a lot more effects in the neurons. What it would mean is if your sodium level, your potassium levels are high, this actually, the concentration difference between intercellular and extracellular are lower. And so what ends up happening is things aren't actually moving as much. So your reflexes may actually be decreased. You may have numbness and tingling. And this is actually based on the muscles, the nerves and different cells not being able to contract practically, or sometimes being overly contracted. Hypokalemia, kalium is potassium in Latin, will actually lead to weakness, fatigue, muscle cramps, and twitching. Again, if this is low, when you're looking at this, you're actually looking at the serum, so it's extracellular. If this is low, potassium will be able to leave quicker, and things aren't necessarily going to act the right way. And when you're trying to reset it, it will be actually harder. And so these ion concentrations will cause effects on the systemic level. Now, I mentioned this a little bit already. You have the functional unit of the nervous system called the neuron. And it is composed of, I'm going to put a circle here to start to think of it as a normal cell with everything in it. And you have projections coming off called dendrites. These have usually ligand-based receptors on it. You have the axon, which is a long projection, and you have vacuoles at the ends. Now you have modified the cell to act a certain way. So you have the dendrites over here, and therefore incoming messages. They actually do have ligand-gated channels which act as receptors. Now some of them are going to be a little different. A lot of times we will deal with sodium channels, but you do have chloride channels and others over here. You have the cell body or the soma. Now remember, anytime you see soma, it means Body. So the soma of the neuron is this body, and it is where the dendrites are attached to, and the dendrites cause regional depolarization, and it starts accumulating, and it's sent to the soma. And once the soma gets uh, pretty much depolarized enough, it ends up causing the entire cell to depolarize, and it causes sodium channels to open. These are voltage gated sodium channels. And then you send the message down something called the axon, which axon is a long projection, and it's attached to the soma by the axon hillock. These actually are usually myelinated, which is a phospholipid bilayer that goes around the cell, which we'll talk about it later, and it sends a message to the vacuoles. Now, one of the things that is different between the neurons and other cells is it actually has a lot of mitochondria due to the energy demands. It means it requires a high level of glucose and a high level of oxygen. If your oxygen levels or glucose levels drop too much, it starts affecting brain function relatively quick. In fact, low levels of oxygen can start killing neurons within minutes, actually within seconds in some cases. And so the thing is, you always want to make sure that oxygen and glucose levels are at the right concentration going there. Mitochondria do tell you that you do require a lot of energy. Just to give you an idea of how much energy it needs, if you look at how much blood the heart beats, about 25 to 33% of every heartbeat goes to feed the brain. And that doesn't change. You constantly need that level. It also has modified rough endoplasmic reticulums. If you remember, rough endoplasmic reticulums have ribosomes. And in the neurons, you have a lot more need for protein-bound, I'm sorry, uh, membrane-bound proteins for receptors and also to make the neurotransmitters. And so you make them in these specialized ribosomes, sorry, these rough endoplasmic reticulums with ribosomes called nysyl bodies. These will be used for vesicles and proteins and dendrites and neurotransmitters. One of the things I will also tell you is always look at the name neuro for neuron, transmitter. It's something that sends. So vesicles will be filled with neurotransmitters at the axon terminae, end of the axon, and vesicles will have proteins at the dendrites. There's other enzymes I will talk about later. Now, when we're looking at the nervous system, we do have a gross division. We can talk about the peripheral nervous system and the central. Now, this is more of an anatomical division because the peripheral and central will go together and messages will be sent in different directions. The reason these are important is because we have support cells called neuroglial cells, which are found in certain areas. So for instance, you have Schwann cells, which myelinate the peripheral nervous system. Myelin is a phospholipid bilayer that goes around the axon. The Schwann cells are the cells that make the myelin in the peripheral nervous system. You also have astrocytes, and astrocytes control the fluid that goes from the blood into the nervous system. It actually makes sure that you have the right ion concentration in the central nervous system. It provides nutrients to the central nervous system, and it also helps repair portions of the central nervous system. And it also controls where the blood should go there. Now, astrocytes are extremely important for the central nervous system to feed this. Now, we also have oligodendrites. Oligo means few, dendrites you already know, but oligodendrites help myelinate the central nervous system. Besides myelination, it does help keep these neurons alive. And we also have macrophages in there. Some of these are called microglial cells. Some of them are periarterial macrophages. What you can tell again is peri means around, arterial, artery. So these are macrophages that are around the artery. And what they're doing is eating anything that may be trying to get into the nervous system, which can do a lot of damage to you. And so you can actually see a lot of these different things with the cells. Now, when you're dealing with cancers, sometimes you may hear of a schwannoma. Schwannoma would be a cancer of the Schwann cells. Astrocytoma would be a cancer of the astrocytes. And so you can actually see how different cells will be used in names. Now, one of the things that happens is neurons can be extremely long cells. If these cells are naked, they lose the action potential under long distances. That's why most of the longer ones tend to be myelinated, and the ones that are not myelinated are usually short. Myelin is made of a special phospholipid, by the way. It's called sphingomyelin. You'll notice that you have the word myelin here. And they're found in glial cells in the glial cell membranes. These wrap themselves around the axon. Now, myelin acts as an insulating sheet, and wherever they actually wrap, the charge doesn't go. It actually doesn't even have sodium or potassium voltage-gated channels, and so the charge will actually jump between little, pretty much little openings in the myelin sheath called nodes of Ranvier, which is where you do have the sodium and potassium voltage-gated channels. This allows the action potential to jump. And so if you think of saltatory, a salt is a jump, so saltatory is jumping. It's jumping from one node to the other. Myelin can also, the other thing it does is it speeds up how fast the action potential, namely because it doesn't have to travel throughout the nerve. It can jump between node to node. And the thing that makes the myelin sheath again is the glial cells. This also helps protect the neurons from damage, and it can also help in healing a neuron. So if a neuron is damaged, if there's enough glial cells around it and it's not in the soma, you can sometimes heal neurons and nerves. Now, one of the things that happens when the charge goes from the dendrites, I'm going to draw the nerve like this with little dendrites coming off here. You'll notice that, yes, this is actually really interesting, nice stick figure. The message comes from the dendrites. You accumulate the voltage, and once you get a threshold, the axon depolarizes, causing sodium channels and potassium channels to open and close. The myelin sheath keeps it going pretty much in a saltatory pattern. And when you get to the end, instead of having sodium and potassium gated channels, you switch to having a voltage gated calcium channel. Over here, you will notice that the calcium inside the cell, intracellular, is a lot lower than the calcium outside the cell. So calcium, like I mentioned before, will move into the cell. That calcium will bind to a specialized protein called calmodulin. And calmodulin causes the vacuoles to move, and so you'll end up having a exocytosis. So cell membrane contains cell surface receptors for neurotransmitter. These can be the ligand-gated sodium channels that I keep talking about. They open and it brings everything closer to resting membrane potential. I put RMP, I meant resting membrane potential. And when you get the resting membrane potential close to threshold, the channel's open. If threshold is not reached, it closes. So if we reach the threshold, the voltage-gated sodium channels open first. It causes a depolarization. Potassium channels then open, causing a repolarization, and that jumps and eventually goes to the calcium channels. Now, one thing, while moving all these sodium out of a cell and potassium into the cell, calcium into the cell, pumping it out, all that letting stuff go through diffusion doesn't require energy, but setting everything up requires a lot of energy. That's why, again, you need the mitochondria. You also need to make neurotransmitters in vacuoles, and most of these neurotransmitters are made by modifying a amino acid. Usually it's tyrosine. And so you make these in vacuoles, so you need to actually make a lot of vacuoles, and that's why you have a lot of these modifications. You'll notice I keep on repeating certain things over again. So what happens is neurotransmitters bind to ligands. Neurotransmitters are ligands. They're actually molecules sent from one neuron to another, and they can bind ligand-gated receptors. In neurons, these will usually be sodium channels. Again, you can see sodium concentration. You open a sodium channel, it's going to allow facilitative diffusion into the cell. This is going to cause the cell membrane to become more positive until you reach threshold. Once you reach threshold, at the hillock, all the voltage-gated sodium channels open, causing all the sodium to leave the cell in that area and reach in that specific area 0 and continue to go over 0. And then when the charge approaches 0, the voltage gated potassium channel is open. You can see here potassium will move back into the cell, and as it's positive charge, it's going to lose positive, so you're going to gain a negative charge. That's your action potential, and it's going to be sent down the axon, one node of Ranvier at a time. And so it's going to jump, that saltatory pattern. Once the action potential reaches the axon terminae, you'll see that calcium channels open. This is important because calcium channels will move in, and that will cause a release. One thing to actually know, at the axon, the action potential is all or nothing. You either get the action potential or you don't. There's no in between, like in the soma and the dendrites. You get it, all the channels open. And at the axon terminae, I actually mentioned this already, calcium enters a neuron via facilitative diffusion. It binds to a protein called calmodulin. If you notice, CAL, calcium, modulin. It's modulated by calcium, and this protein is found on the vacuoles. And if calcium binds to the calmodulin, it causes a cytoskeleton to take the vacuole and move it, so the cell membrane and the membrane of the vacuole fuse, and whatever's in the vacuole gets sent out. If you remember, this is a process called exocytosis. Exo meaning outside, cyto cell or bag. Osis is a process, so it's a process of pushing a vacuole out of the cell. Transmitter leaves, and the transmitter can then bind to a receptor on a neighboring cell. And you have the same message being sent from one cell to the other. One of the things to remember, though, is not all of them are going to be, not all the receptors on the cell surface are going to be excitatory. Not all of them are going to be for sodium. Some of them are actually going to act on chloride channels. And if it acts on opening chloride channels, chloride will actually bring it out farther from threshold, and so it's going to be an inhibitory. So receptors can actually be inhibitory or excitatory, and so it depends on what binds to the receptor and also what type of subtype receptor you have, which we'll talk about in a little bit. Now, I'm going to actually mention a clinical condition, which is pretty much a condition where the glial cells die. It's ALS, amyotrophic lateral sclerosis. It is actually getting harder, and the reason these things are getting harder is you're removing the fat, you're demyelinating. And so what ends up happening is, as this is actually on the spinal cord, you actually initially have trouble with balance. You have trouble moving, walking. Then you actually will start having tripping and falling. Then you'll have weakness of the legs and ankles. Hands will become weak and clumsy. You start getting a slurred speech, and all this, you notice, tends to be a motor aspect. You're actually first of destroying motor nerves. And eventually, though, this continues, and you end up having changes in how you think and how you behave. And so eventually this will lead to other issues, not just peripheral. It is a central nervous system demyelinated disease, but it affects the peripheral because it's a spinal cord. You have other ones. Sometimes people actually do like to do discussions on this. So you do have something called a demyelination disease of peripheral, GB syndrome. And what happens initially is you start feeling pins and needles in your fingers and toes. What that means is the peripheral nerves, the nerves farthest away, tend to stop sending messages to your nervous system. Then you start feeling the weakness in the legs and upper body. You'll notice that there are a lot of similarities. You then have difficulty moving your muscles. Now you have messages going from your brain to your muscles, and so the peripheral nerves, the outer nerve, start actually having the effect because of the nerves that are actually leaving the central nervous system, that are in the peripheral nervous system. You're going to start having vision problems because the nerves, again, are not going to be able to necessarily come in, and you're not going to be able to accommodate and you're not going to be able to accommodate the light. And so you're going to start having inability to move eyes as well and inability to see as well. And then you're going to start having severe pain, achiness, shooting cramps, and usually it's worse at night as you're trying to rest and stuff. This actually affects the peripheral nervous system, and part of it actually controls your bladder and bowels. So those are actually going to be things that are going to affect. And you do have part of the peripheral nervous system, which is part of the autonomic, and so you can end up affecting your heart rate, blood pressure, everything can, and eventually cause difficulty breathing. And so these are actually some of the peripheral, this is an example of a peripheral demyelinating disease. Now, there's a couple different things that actually are important. What keeps neighboring cells from being triggered after a neurotransmitter has been released? Or how do you keep the cell from being continuously activated once you release neurotransmitter? And you have enzymes that can break down the neurotransmitters. Over here you have one, and you can see what the name tells you. You have a monoamine oxidase, and a monoamine oxidase works on tyrosine type neurotransmitters. These are serotonin, norepinephrine, dopamine. And monoamine oxidase will take a monoamine, those three types of neurotransmitters, and break them. And if they're broken, you no longer have that neurotransmitter. It will not affect the neighboring cell. You also have something called a cholinesterase. And the cholinesterase breaks apart a neurotransmitter called acetylcholine. And if it's broken down by this, what's going to happen is the neurotransmitter will not function. By the way, you do have medications that work on both of these. As acetylcholine tends to be used for a lot of movement, if you, for some reason, don't have receptors for acetylcholine, you can get a acetylcholinesterase inhibitor, which means you're going to maintain a higher level of acetylcholine, which makes it more likely to activate the receptor. You have an old class of antidepressants called monoamine oxidase inhibitors. A lot of times they're usually only given on rare cases now, but what it does is it keeps you from breaking down dopamine, norepinephrine, and so it actually maintains those in your system, so you are more likely to, again, activate the receptor. The other way that a cell can keep from being activated too much is on neurons. The receptors for neurotransmitters can be there's actually let me say that again. On neurons, there's receptors for neurotransmitters that don't trigger the cell to contract, but pull the neurotransmitter back. So if you have enough neurotransmitter in between two neurons, or between a neuron and a neuromuscular junction, you can reabsorb the neurotransmitter. This is actually seen on something like a neurotransmitter like serotonin, where you have special medications which inhibit the reuptake of serotonin. If you inhibit the reuptake of serotonin, you maintain more serotonin between the neuron and its target tissue. And so what ends up happening is you have more serotonin. It tends to activate those receptors more. These are used in some of the antidepressants, selective serotonin reuptake inhibitors, for instance. You also have receptors on target cells that can be brought into the cell via endocytosis. So if you're constantly activating a cell, the cell that's being activated can pull its receptor in through endocytosis. Now, are there ways that we can get these speeds to increase? And there are two things we notice that can increase speed of transmission in a neuron. Remember, you're sending mainly messages through the axons. So one of the things we do is we can myelinate it. So we can have myelin around our neurons. And as that acts as an insulation, that means that the cells will actually send the message faster if it has myelin. If it doesn't have myelin, it'll be slower. You can actually see this with two different types of pain. Now, this is a generalization, but it does work for this case. If you have a dull aching pain, it tends to be a more slower pain, a pain that gets to you slower. And so the dull aching pain usually tends to be more of a naked neuron. It doesn't necessarily have as much myelinate. sharp pain or burning pains tend to be quick. They tend to be myelinated, and so that speed allows messages to be sent a certain way. The other way we can increase the conduction is by increasing the diameter of the axon. What I mean by that is if we have a small diameter axon and a big diameter axon, diameter being the space between one side of the cell to the other, here or here, the bigger one will move the charge faster. Now, for us to make bigger axons, we need to actually build up what's inside the cell. We actually need more cytoskeleton. So for us to make our nerves work faster, we have to build up the proteins, also the phospholipid, and we have to make sure we have the myelin. So we need the proper lipids to be in the system so we can actually build the axons bigger. And so, yes, we can increase the speed. So if you actually want to increase the speed of your axons by movement, you do need to make sure you can use those nerves and give them time to rest to build up the axons. Now, as we're dealing with neurons, we actually do have two pathways we can talk about. One of them is a descending pathway, which means it's going from the brain to a target tissue. The other is an ascending, which is moving from a target tissue, some sort of receptor up. Now, when we're actually looking at this, we usually talk about two types of target tissues. It could be another neuron or something else. Now, if the other tissue is a neuron, we end up having something called a synapse. What a synapse is it's a connection, highlighted here, it's a connection between one axon terminae and the dendrite of another neuron. And so that's where the synapse is. And that's where you're going to send neurotransmitters, and it's going to be very specific and very tight, allowing for quicker responses to the release of neurotransmitters. Remember, neurotransmitters reach the receptors, the ligand-gated receptors, based on diffusion. So the closer everything is, the faster this will occur. Now, we also have another one, which we can usually talk about it as a neuromuscular junction. Usually this means you have a neuron and muscle, and so you have the neuron terminate coming in contact with muscles, but it can also hit glands to cause different things to be sent. Now, while the axon is where the action potential goes and it's an all or nothing, one thing to remember that with neurons, once you pass the dendrites, it's an all or nothing. And the interesting thing is it can be an additive effect. What I mean by that is you can end up having multiple axon terminants, axon A, on different dendrites, meaning that you can actually have multiple neurons acting on that. And that's how it actually can get to threshold. So in the dendrites, it's an axon hillock. It is this depolarization tends to be additive. Once it gets to the axon itself, it is all or nothing. Now, not all neurotransmitters take the membrane closer to threshold. You have two types. You have excitatory postsynaptic potentials, EPSP, and inhibitory postsynaptic potential, IPSP. And as these names suggest, these action potentials, these synaptic potentials can either bring things closer to threshold or away from it. Now, when we're out dealing with neurons, another thing to actually mention is you have the first neuron sending a message to the second neuron. And I'm going to draw it like this. You'll notice that I'm no longer drawing the dendrites. When we're actually drawing to show the pathway, a lot of times we're only using the soma and the axon so we can see. So the synapses between these two. And so we can talk about the first neuron being presynaptic, pre being before synapse, and the second neuron being postsynaptic. And then you'd have some sort of target tissue here, another target tissue. And so we can have multiple neurons converging onto another neuron. So you can have excitatory neurotransmitters and inhibitory neurotransmitters. An inhibitory will move it farther away from threshold. And again, like I mentioned, this is usually due to chloride channels. Chloride channels open, chloride comes in, and it tends to make stuff more negative. And so what can end up happening is you can have two neurons firing, and the negative will inhibit the positive, so it won't necessarily fire. But you can also have multiple positive, uh, well, pretty much excitatory postsynaptic potentials, which get this neuron closer to threshold. And so that's why you have excitatory postsynaptic potentials and inhibitory. The excitatory postsynaptic potential, the neuron is actually causing the next cell to become more positive, getting it closer to threshold. And in inhibitory, it's getting farther away from threshold, meaning you're inhibiting the response. And both of them are important. Now, I do want to mention the somatomotor tracts. Somato means body, motor, movement. So these are the tracts that are causing movement. And you have two neurons. The first one is referred to as an upper motor neuron. And you'll notice that I put UMN here, which means it's the upper motor neuron, just initials. And it sends the action potential down the axon, releases a neurotransmitter. The neurotransmitter that the upper motor neurons use is called acetylcholine. And you'll notice that I'm writing here ACH because a lot of times when I'm talking about acetylcholine, I may say acetylcholine and I'll write ACH, but it is still acetylcholine. Acetylcholine binds to a receptor, and this receptor in the somatomotor, upper motor neuron, the receptor that the upper motor neuron binds to, so what you're talking about is the first neuron coming down, is going to release acetylcholine, and it's going to bind to a second neuron. That second neuron would be the lower motor neuron. The reason we call it lower motor is because it's lower from the brain. So acetylcholine will bind to a receptor, and the receptor will be here on the lower motor neuron. And acetylcholine usually is a ligand for sodium ligand-gated receptors, which means that will bring it closer to threshold. And if you have enough firing, the second motor neuron, the lower motor neuron, will fire, and it tends to send neurotransmitter to a target tissue, which is muscle. And in the muscle, where the neuron and the muscle come together, we have an area called the neuromuscular junction. And at the neuromuscular junction on the muscle, you have something called a motor end plate. And this motor end plate has receptors for acetylcholine. And they're specialized motor end plates, they're specialized receptors that don't just bind acetylcholine, but bind to nicotine. And so we call them nicotinic receptors. So these receptors, even though they're made for acetylcholine, something like nicotine can bind to them. And so you can activate these nicotinic receptors by and you can end up having contraction. Now, if you do not have these receptors, the acetylcholine nicotinic receptors technically you have it on the neuron here on the surface and on the target tissue. If you don't have the nicotinic receptors, you cannot have movement. And there is a condition where you make an immunoglobulin to neuromuscular junctions, killing them off, and you end up losing the ability to move eventually. We can usually give immunosuppressants and acetylcholinesterase inhibitors to keep you able to move. Now, there is another thing, and you will talk about this in micro. You have a bacteria called Clostridium botulinum, and it produces something called botulinum toxin, A, B, and botulinum toxin B can inhibit the release of acetylcholine. If you do not release acetylcholine, what you're not going to be able to do is move. So what you end up with is a paralysis. You become paralyzed, and it's a paralysis which is flaccid, meaning it's just limp. Now, besides the neuromuscular, the somatomotor, we also have autonomic. Autonomic is another two neuron system, and a lot of people talk about, it's a peripheral. It does have parts that are central and some that are peripheral. It's a two neuron system. And so the neurons, you have the upper motor neuron the first neuron sending messages down, upper motor neuron, and that is going to release acetylcholine, ACH. If you release acetylcholine in the synapse, it's going to go to the synaptic cleft where it can bind to receptors potentially. And if it binds to a receptor, this acetylcholine from the sympathetics will actually end up causing the cell to fire. And so you have the sympathetic here. You also have the same effect on the parasympathetic. They're both going to use the same acetylcholine, and you're going to have nicotinic receptors on the postganglionic neuron. And this two neuron system, if you notice, is working so far like the somatomotor. The neuron sends acetylcholine, it goes to the synapse, you have nicotinic receptors, which acetylcholine binds to. What happens next depends on whether you're looking at the sympathetic or the parasympathetic. So the first part of this, whether it's somatomotor, the autonomic sympathetic or autonomic parasympathetic, all of them will work the same way. You're releasing acetylcholine, and the acetylcholine will bind to the nicotinic receptor on the postganglionic neuron. Now, you'll hear postganglionic and preganglionic neurons. What it has to deal with is this. You do have ganglion, which is a collection of cell bodies. Technically, the preganglionic is before you get to the cell bodies here. Postganglionic are once you leave it. Ganglion is a collection of cell bodies, so you usually have a bunch of neurons coming here. Now, for the parasympathetics, I usually like to talk about it first because what you have is you have a neuron, and like I said, the first neuron, the upper motor neuron, is going to release acetylcholine, and it's going to bind to nicotinic receptors. These nicotinic receptors are going to trigger sodium channels to open, and it's going to eventually cause, if you have enough, an action potential. And in a parasympathetic, you release acetylcholine again, ACH, between the lower neuron and the target tissue. Acetylcholine will leave, and it'll bind to a different type of acetylcholine receptor called a muscarinic receptor. Now, muscarinic receptors are also triggered by a poison called muscarine. And these muscarinic receptors are usually a G protein receptor. And I'll mention exactly what a G protein is. But what ends up happening is muscarine receptors have a lot of different types. They all bind to the chemical muscarine, which are found in some poisons. And you have two different things that can happen. You can bind to what's called a GQ receptor, which is stimulated, and it produces a couple molecules like DAG and IP3, don't worry about that, to activate something called a protein kinase C. Now remember, kinase means that you're going to take a phosphate from usually an ATP and put it on something. In this case, you're going to phosphorylate a protein. Now these proteins activate cells to do something. There usually are the subtype M1, M2, and M5. I'm sorry, M1, M3, and M5. That's not M2. It should be 3. So this, don't look at 2. This is 3 and M5, so odd. And these are going to cause things to become active. Now, these are causing the cells to be active, but when the cells become active, they can actually cause something to be relaxing. So with these muscarinic, if they're GQ, they are excitatory, and what they do is they can produce mucus secretion. They can cause cells to divide. They can cause things to be pretty much certain hormones to be secreted, certain messages to be secreted to get the body to relax. Now these are all G protein receptors. And what a G protein is, it's a protein that deals with something called a GTP, a guanine triphosphate. And when it's activated, the GTP comes in, it causes the protein to turn on, and it ends up causing this huge cascade of events which will eventually activate the cells to do something, to move. And these are actually found in different parts of the body. So for instance, you have some smooth muscles that use these, and they will actually constrict. And so in some blood vessels, if the G protein is activated, it will actually cause constriction to move blood to others. In some, it will actually cause G proteins to a G protein, a GI, or I'd call it a GI, not a GO, but you can either one. These actually inhibit. And so you activate this, and it causes ion conductions to change. It causes chloride to go in, and so it pulls it away from threshold, causing these things not to activate. And so in some cases you have this, and it causes muscle relaxation. And these are M2 and M4. So if you notice, odd are usually, the odd muscarinic are going to be things that activate. The inhibitory tend to be even receptors. Works out pretty nice. Now, with sympathetic, we have another type of receptor. So we're going to have the first neuron. It's going to send acetylcholine. The acetylcholine will bind to the second neuron, the lower motor neuron, on a nicotinic receptor, and this neuron will send a different type of neurotransmitter called norepinephrine. You will a lot of times see, when I'm actually writing this out, me write norepi for norepinephrine. It is also a same receptor or similar receptor to epinephrine. And so these are called adrenergic receptor for adrenaline. And what they're going to do is they can actually either activate things or deactivate, depending on what type of receptor they are. Usually we talk about the sympathetics as activating the system. And overall, when we see it systemically, we see an activation. And so usually the things that you'll notice as activation are going to be the beta receptors. You'll see I put B1 and B2. So B1 receptors work on the heart. And if the norepinephrine leaves us to the target tissue, acts on a B1 receptor, it's going to cause the heart to contract faster and stronger. And that causes an increase in contractility, strength, and blood flow. The speed and how much blood gets pumped out increases, and so you actually have an increase here. This is epinephrine. A lot of times people talk about the fight or flight, but think of fight, fly, or fright. So you can either fight, you can run away, which is a good thing, or you can be scared. These will increase your contractility, so you can actually run get all the nutrients and everything you need where it needs to be to run faster. So B1 will increase cardiac output. B2 is interesting because it's an inhibitory, and it's inhibitory to smooth muscle. And it's inhibitory in certain smooth muscle, I should mention, namely the ones that actually cause constriction of the respiratory tract. So it opens up the respiratory tract. It causes decreased secretions. So you don't want to be having a lot of secretions if you're trying to run away from a bear. It ends up causing you to be able to get more blood flow to an area in some areas, and you'll actually end up having more airflow come in. So it gets you everything you need for respiration and for energy. Now I talk about the beta first because it shows you more of the sympathetic, but you also have alpha receptors. And alpha receptors do work on smooth muscle. A1, for instance, causes some smooth muscles to contract. So the smooth muscle in your periphery usually contract, pulling blood away from the skin into the organs inside the body. Alpha 2 also causes a release of something called glucagon. Now glucagon is a hormone, which I'll talk about in the next lecture, which glucagon is released from the pancreas when you have low glucose levels, and it causes more glucose to be released in the blood so you can use it for energy. It actually comes from the pancreas. It also causes the contraction of the gastrointestinal sphincters to make sure that food isn't moving through the intestines at the time. Everything in the intestines stops because you don't want to be trying to digest and move blood to different areas when you're trying to run. It also increases thrombocyte aggregation. A thrombo means clot, cyte means cell. And so you have this, so if you're cut when you're scared, you can clot immediately so you don't leave trails of blood. It does also inhibit insulin release, and insulin is released to get sugar levels to drop from the blood, and so this actually causes the blood sugar levels to increase rather than decrease. Now, these are things to know about the neuron system, and I will get the endocrine system coming up soon. But you have a resting membrane potential. This resting membrane potential is caused by the sodium-potassium ATPase. When you actually want to send a message, you have to allow channels to change. So this resting membrane potential is caused because you're actively pumping sodium out, potassium in. Know the sodium-potassium ATPase. The action potential is caused by sodium and potassium moving. Now, as you've been pumping sodium out and potassium in, when you want the action potential, you open the sodium channels and you have facilitative diffusion of sodium. You open the potassium channels, it allows facilitative diffusion of potassium. This is based on concentration difference inside and outside the cell. Now, we talked about the neuron, very important. You have different parts. You have the dendrites, the soma, and in the soma you have the axon hillock and the axon. Now, the dendrites receives the messages, the dendrites receives a message, and the depolarization of the dendrites by receptors tends to be additive, and it isn't all or nothing. You get to the soma, that's where you accumulate the depolarizations, and you send it to the axon hillock. Once it gets to the axon, the message is sent all or nothing. It goes through the axon and you have the myelin sheath to help the axon send the message more, but not all cells have myelin sheath. Small neurons may not have myelin sheath, and pain receptors tend to be not as myelinated as others. So you have slow and fast because of that. The axon terminae is the end, and that's where you have the vacuoles, where they actually, when the action potential gets to the axon terminae, remember that you have sodium channel, remember that sodium channels are not used there. It's actually the calcium channels. So calcium comes in, binds to the calmodulin, which causes the neurotransmitter to leave. Remember that there's a division of the nervous system. You have central and peripheral, and it's just telling you, is it from the brain and spinal cord, or is it outside? You do have glial cells, and there's a lot of glial cells you want to actually make sure you know and know what they do. For instance, know that the microglial cells are macrophages that eat things that aren't supposed to be there. Know that the astrocytes form the blood-brain barrier. Know this, pretty much the difference between the Schwann cells and the oligodendrites. I also talked about a couple neurotransmitters. There's more. Right now I talked about acetylcholine and norepinephrine. Norepinephrine is a monoamine, and you have a lot of other ones. Acetylcholine is, if you notice, it's a main excitatory in the peripheral. When we talked about central, you'll see a couple more. Also know that there are receptors. You have different types of cholinergic receptors. Cholinergic is for acetylcholine. You have the nicotinic type and the muscarinic. Nicotinic will bind nicotine as well, and you'll notice that the nicotinic type are in the pretty much a somatomotor, the autonomic sympathetic and autonomic parasympathetic. You also have adrenergic receptors, and the adrenergic receptors, you have the A1s, which affect arteries, A2, which affects blood, B1 that affects the heart, and B2 that affects the respiratory tract. They do affect other things, but if you actually understand how it affects these, you understand how it works. You can also see why these will affect different things. You do have the subtype receptors. For instance, if it's a GS, it stimulates, and it usually works with protein kinase C. GI tends to be inhibitory. You have the descending tracts. So we have somatomotor, which you have an upper motor neuron and a lower motor neuron. You have the autonomic parasympathetics, which have the preganglionic and the postganglionic, and the autonomic, which is, again, pre and postganglionic. I hope you enjoyed this lecture, and have a nice day.